All right, so the second mechanism we have for summing PSPs at the postsynaptic cell is temporal summation. And to illustrate temporal summation, we're going to take a look at this figure right here. What we are doing is we're recording basically at the trigger zone of the postsynaptic cell in response to action potentials that are coming in from the presynaptic cell. Okay, excuse me. Okay, so imagine that you have two action potentials coming in from the presynaptic cell, and they're spaced a little bit far apart in time. That's what's shown here on these axes, right? So these little carrots or arrowheads, whatever you want to call them, these are when the presynaptic cell is having its action potentials, right? So we have one action potential that's going to cause a PSP in the postsynaptic cell. We have a second action potential that's going to cause a second PSP in the postsynaptic cell. Notice that these PSPs are both sub-threshold, right? So we get these PSPs, but we don't get an action potential because we are not depolarizing the postsynaptic cell enough to trigger an action potential, right? However, imagine what might happen if you were to apply those two action potentials in the presynaptic cell closer together in time. What do you think would happen with the PSPs here? Take a moment to think about that. Answer that question. Okay, so hopefully you figured out if you have your action potentials happening close enough in time in the presynaptic cell, that's going to lead to two ePSPs close together in time in the postsynaptic cell. And if they occur close enough in time, they will be able to sum. So that's what's happening here. They're summing, right? Remember that graded potentials can sum. So they're going to sum. And in this case, they've summed to bring the cell to threshold, and we get an action potential. All right, that is the phenomenon of temporal summation. So basically that's summation of PSPs occurring close together in time. All right, so finally, not finally, I don't know, finally, I just wanna kind of put this back in context for you, okay? So we've looked at these phenomena of spatial and temporal summation in kind of these isolated examples, but I want to remind you that neurons are forming all of these very complicated networks, right? And so every neuron is receiving, well, unless it's at the very beginning of the pathway, it's receiving many inputs from many cells. It's kind of constantly being barraged with all these inputs. It's doing spatial and temporal summation all of the time, okay? And so like spatial summation, temporal summation, they kind of don't really occur in isolation. Really what the postsynaptic cell is doing is kind of a combination of both because it's just getting so many inputs all of the time, okay? All right. Okay, so I wanted to include this slide because students sometimes have trouble stringing together all of these events that we've gone over with action potential generation, synaptic transmission. So I wanted to just kind of do, sort of walk you through this to clarify what happens as a signal goes through a network of neurons, okay? So we're gonna start here, we're gonna like generate a signal up here and watch what happens to it as it moves through this network of neurons. Okay, so we're gonna start up here. First, we get a graded potential in the dendrites and cell body of this neuron, okay? Next, that graded potential, those graded potentials are going to come to the trigger zone, they're going to sum, and if you reach threshold, we're going to get an action potential. if we don't reach threshold, nothing happens. But let's just take the example of reaching threshold. Okay, so let's say we've reached threshold. What's going to happen is we're going to generate an action potential. That action potential is going to travel down the axons of this neuron. This neuron has a branching axon, so it's going to travel down the axon. Once that action potential gets to the axon terminal of that neuron, we're going to get synaptic transmission, right? So we're going to open our calcium channels. Calcium is going to trigger exocytosis of neurotransmitter, okay? So that's what happens here. Synaptic vesicle release, really it should say neurotransmitter release from the presynaptic neuron. That neurotransmitter then gets detected on the postsynaptic cell, and that leads to our PSPs, our postsynaptic potentials, which are graded potentials, okay? Now, that graded potential in this cell is analogous to the graded potential we saw up here, right? So now we're going to go through the same series of steps. We have a graded potential here. It's going to reach the trigger zone. We're going to get an action potential, assuming that we reach threshold. That's going to travel down here. We're going to get synaptic transmission down here, and so on down the line, okay? So I just wanted to kind of put that into context for you. Some people have trouble like putting it all together. Okay, finally, I just want to mention that we have this lovely video for you to watch about synapses and synaptic transmission. I encourage you to watch it. Very, very nice animations. One really nice thing about this animation is you can really see the way that postsynaptic potential is like spreads from the point at which it's generated. So it'll start there and spread and degrade over distance. Just like that pebble being thrown into the pond. It has a very nice animation of that. So yes, please watch that video. That concludes our discussion of integration of inputs and of synaptic transmission in general. These are your learning goals. Here's your vocabulary. Enjoy.